am Mixão. I am from the National Institute of Health of Portugal, and I'm here to talk about inter-pipeline cluster congruence assessment that we performed at European level. So whole genome sequencing is a powerful ally for the surveillance of bacterial pathogens. However, different laboratories employ different bioinformatics pipelines, and this casts doubt on the comparability of their clustering results. So, taking advantage of a large European consortium involving multiple European countries from different sectors, so the clinical sector, the food safety sector, the animal sector, we performed an evaluation of the cluster congruence between the pipelines they use in the routine genomic surveillance. For this, we collected a data set with more than 2,000 isolates for 4 important foodborne bacterial species: Listeria, Salmonella, E. coli, and Campylobacter. And we gave these data sets to each laboratory and we requested them their clustering results. Afterwards, we did pairwise comparisons between the different results. And we did these assessments. What we found was that there is a general high concordance between the allele-based pipelines for all species, except for Campylobacter jejuni, where the different resolution power of the different schemas in place generates very different results between laboratories. Moreover, we found that there is some backwards compatibility with the traditional typing data, like sequence type or serotype. And we also found that in concordance with previous studies, there is some difference in the diversity of different traditional typing groups, such as there are some sequence types that are more clonal than others, and some of them are also more diverse than others. And this is useful information for outbreak case definition, as we cannot treat all of them in the same way when we are trying to define an outbreak. And regarding outbreaks, an important question we had was whether the different pipelines actually are able to signal the same outbreaks. So if different countries employing their different pipelines but provided with the same data sets, they would raise the same outbreak signals. And what we found was that if we use static thresholds to determine the outbreaks, as it is commonly performed, we find some incongruencies between the different countries. However, if we flexibilize this threshold application and take into consideration the specificities of each pipeline, we increase the chance that 2 different laboratories signal the same outbreaks. And what is the practical impact of these results? So every time there is an outbreak at European level, and that is of concern, there are some queries that are sent to the different laboratories, and these laboratories are requested to send for a centralized database all isolates of their collection that fulfill certain criteria. And these criteria are usually a static threshold of distance to a representative isolate of an outbreak. And in practice, what our results show is that if we do not flexibilize this threshold and do not take into consideration the specificity of the pipeline of a given laboratory and that it is different from the centralized pipeline, we may be not reporting isolates that actually, when analyzed by the centralized pipeline, would belong to this cluster. In the end, this study opens good perspectives regarding interlaboratory communication and cooperation and raises awareness about the need of flexibilizing this concept of outbreak case definition.